All right. So our next presentation is, the title is The Effect of Red Spectrum LED Lighting on Commercial Turkey Breeder Hen Performance. And our presenter is Clara Ziezold from the University of Guelph. Clara, if you're ready, take it away.
Thank you, Val, for that introduction. Can you see my slides?
Yep, they look great.
Perfect, thank you. So yes, I'm Clara Ziezold, and I'm a master's student under the supervision of Dr. Gregor Dedekeritz at the University of Guelph. And I'm looking forward to presenting our data so far on our field trial on the effect of red spectrum LED lighting on commercial turkey breeder hen performance. So when we're talking about reproduction in birds, photostimulation is a pretty good place to start, since that's the light-dependent reproductive response. In turkeys specifically, which are long-day seasonal breeders, that occurs through the exposure to increasingly shorter nights and longer days. And that does two things to activate the reproductive axis. So firstly, we have the lifting of inhibition, and that occurs through exposure to shorter nights. We have reduced melatonin production by the pineal gland, which downregulates gonadotropin inhibitory hormone. Simultaneously, we have this directly stimulatory effect of the long day, and that occurs when light penetrates the skull and neural tissues to directly stimulate those deep brain photoreceptors of the hypothalamus and upregulate gonadotropin releasing hormone. Gonadotropin releasing hormone stimulates the anterior pituitary gland to secrete gonadotropins, luteinizing hormone and follicle stimulating hormone into systemic circulation, which promotes steroidogenesis at the level of the ovary. So that's where we see that increased estradiol and progesterone production. So since in poultry, reproduction is a light-dependent response, the lighting management of breeders is incredibly important. And if you're familiar with turkey breeders, the light intensity is actually an important part of the lighting program. However, actually measuring light intensity can be a point of contingency. So I just want to clarify some of the terminology and how that relates to reproduction. So most of us are probably familiar with the unit lux. That's lumens per meter squared, or foot candles, which is the non-metric unit. And although in industry and traditionally in research, we use lux as a measurement of light intensity, technically it's actually a measurement of illuminance. And what illuminance quantifies is visible light energy. So lux specifically is calibrated according to human visual perception, so according to this function. And we can see that that's not all that relevant to poultry in general, because their perception of light is far different than ours. So if we calibrate illuminance to the avian retina, so according to this function, that's clux or Galilux, this can be very useful for measuring behavior, since that's the visible light energy to the avian retina. However, as I mentioned, when it comes to photostimulation, that's light perceived by the deep brain photoreceptors, and therefore the eye or retina is not all that important. Instead, what's more relevant is intensity in watts per meter squared. So that's quantifying actual light energy rather than that visible light energy, making it useful for avian reproductive measures, since we're measuring the amount of light that can reach those deep brain photoreceptors. So from now on, I want to focus on these parameters of light and how they relate to reproduction. So we have photoperiod or day length, which is typically measured in hours. So that describes the duration of available light energy. We have intensity in watts per meter squared, is the instantaneous amount of available light energy per surface area. And then when it comes to spectrum, which is measured in wavelengths by nanometers, or when we're talking about the visible light spectrum, that's color, we're really talking about tissue penetrability. So certain wavelengths of light have greater tissue penetrability than others, making them more able to penetrate the skull and neural tissues to reach those deep brain photoreceptors. And therefore, all of these parameters of light can describe the ability of that light to stimulate deep brain photoreceptors. Now, like I said, the spectrum can relate to tissue penetrability. And in fact, red light has the greatest tissue penetration power of any other light spectra. So specifically, red light is 30 times more effective than green light and 1,000 times more effective than violet light at reaching the avian hypothalamus. And for this reason, it's been known since the early 20th century that housing poultry under red light accelerates their sexual maturation and can actually also increase their egg production. And that's in duration of the laying cycle, so persistency, as well as increasing that peak egg production. However, interestingly, this hasn't yet been reported in turkeys, and it warrants investigation because turkeys are not only highly photoperiodic, but also have poor reproductive performance. So that brings us to this trial, in which we hypothesize that due to its greater tissue penetrability, red light activates a larger population of deep brain photoreceptors, resulting in a more pronounced activation of the HPG or reproductive axis. And therefore, our aim was to determine if red light improves reproductive performance in turkey breeder hens in a commercial setting. So to test this, we had 2 light treatments. So our control corresponded to the existing lighting fixtures in the commercial barn. So that was a 61-watt white tube fluorescent light, which I'll denote as W. And our treatment light corresponded to a 10-watt Agrilux 60% red spectrum being 60% red, 20% green, and 20% blue LED light. So we'll denote that as R. And this figure is the light spectrum outputs of each of these lighting treatments as measured by a spectrophotometer at bird level. So what that is showing is intensity per wavelength. So essentially what we're seeing is in our white light control treatment, the majority of the energy output is in the green, yellow, and orange spectra. Whereas, of course, in our treatment lighting, the majority of the spectral output is in the red spectrum. In terms of experimental design, we had 2,650 male line great grandparent hybrid turkey breeder hens, and they were housed and managed under commercial conditions. So all of these hens were housed in a single barn, which was divided into 3 equal light-proof sections. And sections 1 and 2, or R1 and R2, were installed with the red treatment lighting. And each of these pens, so 4 pens per section, housed the same number of birds for a total of 1,060 hens. Section 3, on the other hand, is where we maintain that existing control lighting. We had 2 pens of hens for a total of 530, since we also had toms housed in this section. And in the turkey breeder industry, males and females are kept separate. So since this was performed under commercial conditions, we had a staggered photostimulation between these sections. In sections R2 and W, birds were photostimulated at 30 weeks and 6 days of age. And in R1, they were photostimulated 3 days later at 31 weeks and 2 days of age. So because of this age discrepancy, we're going to, or we have adjusted all of our data in terms of weeks post photostimulation rather than age. And in R1 and R2, the birds were acclimatized to the red light treatment 4 weeks prior to photostimulation. And prior to that, they were housed under the control lighting. So photostimulation occurred with a 13.5-hour photo period and was adjusted accordingly. We have up to 11 weeks post photostimulation of data since this trial is still ongoing. In terms of data collection, we had daily egg production and egg quality measurements. So that included cull egg parameters as well as egg weights at 3 time points. That was by 100 random eggs per section. We also had weekly blood sampling from 10 random birds per pen to quantify plasma estradiol using a commercial ELISA kit. And that was performed at the time of photostimulation and 6 weeks after. And all data were analyzed in SAS with a 1-way ANOVA and 2-piece adjustment. So in terms of results, we did see the expected increase in egg production under red light. So specifically in R1 and R2, we saw increased egg production at 4, 5, and 7 weeks post photostimulation. And that translated to a higher peak than the controls. Overall, up until this point of 11 weeks post photostimulation, that translated to a 1 to 2 egg gain per hen housed in red 1 and red 2. Interestingly, looking at this plasma estradiol, this captured the sexual maturation, and unexpectedly, we saw no differences in profiles between groups. However, looking at the egg production data, that's exactly what we'd expect, since the entry to lay between groups was not different. And differences started to occur around peak production. So it's possible that if there were differences in estradiol, specifically higher concentrations under red light, we may not have been able to capture that. We also saw heavier egg weights in section R1 in the entry to lay. And we don't attribute that to lighting per se. That's most likely an age effect, since those birds were photostimulated 3 days later than R2 and W. But that difference didn't persist over time. In addition, we saw no differences in weekly body weights up until the point of photostimulation. We also saw no differences in fertility and hatchability, nor colleg parameters, or cumulative mortality and euthanasia. So in conclusion, we did see the expected increase in egg production under red light in turkey hens through an improved peak egg production, as has been reported in other poultry. And since this trial is still ongoing, we're interested to see if we see any effects on laying persistency. And this field trial certainly performs or provides a platform for further investigation into the physiological effects of red light in turkeys. And I'd like to thank my advisor, first and foremost, Dr. Gregor Beta-Carrots, as well as my colleagues for their support, advice, and feedback. I'd also like to thank Hybrid Turkeys and Hendrix Genetics for their provision of birds, facility, and resources, especially Nicole and the R&D team. I'd also like to thank Sarah, Yvonne, and farm staff, and a big thank you to Alex Thies, electrical distributing company, for his in-kind contribution of the 60% red lighting. Thank you for your attention.
Great job. Thank you for your presentation. If you've got any questions, folks, please add them to the question and answer box. We have a few minutes. I just have a question about, are these light bulbs commercially available, or are they still being tested? And if they are available, did you do, I guess it might be too early because you haven't finished, but is there a plan to do an economic analysis in terms of refitting your barn with light bulbs?
Absolutely, that's a great question. So yes, these lights are commercially available, and they've been tested in broiler breeders and layers primarily. So we just took those bulbs that have been specifically designed for chickens and tried to see what would happen if we installed them in a turkey breeder barn. So yes, they are commercially available. An economic analysis would also be great, since we're comparing such a high wattage bulb, like tube fluorescent is still quite prominent in the turkey breeder industry. So LEDs, whether they're red spectrum or not, are going to save a lot in energy, as well as any egg production gains.
For sure. Good. Another question here is, how did you decide on the light intensity used in this experiment?
So we initially performed a pilot trial in the same setting, and we started with a much lower intensity, what would usually be used in a layer barn in terms of lux. But we found that the egg production was decreased at that point, so we decided to double the intensity there for this trial. So we suspect that turkeys might need or require higher light intensity for reproduction. So we doubled that intensity, and if we're measuring that intensity in watts per meter squared directly under the light source, that red spectrum LED light is very comparable to the white tube fluorescent. So they were somewhat matched in terms of watts per meter squared. However, that's something we're interested in looking in further in an experimental setting. We actually want to look at the effect of light spectrum and intensity, both combined and on their own.
Yeah, some fine tuning to see if that's necessary. Yeah, for
Exactly.
sure.
Yeah.
One more question here. Is it possible that the light intensity is relatively low to stimulate the HPG axis and resulted in no differences in the plasma estradiol? So just trying to understand what happened, or was it? No, we'll go with the question.
That's a great question too. So when we look at red light treatment in other birds, specifically chickens, for example, we see a higher peak in estradiol under red light treatment. However, we're looking at a bird that's very, very inefficient. These are great grandparent male line turkey hens. So it's possible that they were actually limited, or that we were limited in that sense, that in the number of small white follicles potentially didn't have the capacity to reach much greater than what we saw. But
Perfect.
that's something for further investigation as well.
Further investigation for sure.
Yeah.
Well, thank you so much for your presentation, Clara, and